From the American Neurological Association, I am Ifra Zawar, and this is ANA Investigates. Today, we will focus on the latest in the research on ALS. ALS remains a devastating disease, but the field is now at an inflection point, with a growing pipeline of precision therapies, trial-ready sites, and a new generation of clinician scientists. Yet, barriers persist. There is disease heterogeneity, limited trial access for patients, and rising development costs. For an update on ALS clinical trials and the Healey ALS MyMatch program, a centralized platform that matches participants to trials, we spoke with Dr. Suma Babu. Dr. Suma Babu is an associate professor of neurology at Harvard Medical School and co-director of the Neurological Clinical Research Institute at Mass General Brigham. She was interviewed by Dr. Ryan Jacobson, an associate professor of neurology at Rush University. Here is Ryan.
Well, our topic that we're going to discuss together today is ALS. And in the year 2026, ALS remains one of the most devastating diseases that we face in neurology. So I'm excited to learn more about ALS clinical trials and what we are doing to help these patients down the road. Welcome, Suma.
Thank you so much for having me today.
What do you think are some of the most daunting challenges in the landscape of ALS clinical trials right now?
We have some therapies that slow the disease progression to some extent in ALS, but there's clearly an unmet need for more effective therapies. ALS remains a universally fatal disease. The good news is that we are really at an inflection point in drug development for ALS. There are lots of drugs in the pipeline, many with great targets and precision approaches, and we have the tools to design and run clinical trials. We also have clinical trials ready sites and researchers to conduct clinical trials research. ALS is becoming a popular field for next generation clinician scientists and clinicians to pursue as a career option, which is really wonderful to see. But there are barriers we need to overcome to see therapeutic breakthroughs in this field. There are disease biology related factors. ALS is a complex disease and it is heterogeneous. There are trial access and demand supply factors. There are about 30,000 people living with ALS in the US, and we have only about 400 ALS centers to care and provide research access to all. And there are financial, particularly related to rising research and development costs that many drug developers and small biotech companies struggle with. There's a big need for large-scale sustainable funding and infrastructural support for end-to-end clinical trial development in ALS. If we take the example of oncology, where therapeutic breakthroughs were accelerated in the early 2000s, one of the main reasons for all those success was really the National Cancer Institute supported nationwide clinical trials network. That was a federal support that brought in over 3,000 sites within that network, and that played a huge role in changing standards of cancer care over the past 2 decades. I think literature reports that there have been over 160 positive phase 3 clinical trials conducted by the Cancer Network, and more than 85% of these trials help to change cancer care guidelines.
I understand, and I'm excited to learn a little bit more about what we are doing to change the picture of ALS research right now. Can you tell us a little bit more about the Healey ALS MyMatch program that you are leading? Broadly, what is it and how does it work?
The Healey ALS MyMatch is a clinical trial program in ALS. This is led by the Mass General Brigham, Sean M. Healey, and AMG Center for ALS, with the support of the Neurological Clinical Research Institute that functions as a full-service academic research organization. And we are leading this program in very close partnership with the NEALS Consortium for ALS. The idea behind MyMatch is simple. These are all phase 1b/2a clinical trials in ALS. In phase 1b/2a clinical trial space, there are certain questions we need to answer in drug development pipeline about target engagement, about finding the right dose, early biological signals, safety tolerability. So each MyMatch trial is designed around the science for that specific therapy while taking the advantage of an efficient, centrally located data and operational infrastructure that's built to handle almost anything, from small molecules to monoclonal antibodies, antisense oligonucleotides, cell-based therapies, gene therapies, or even device-based approaches. It could be a placebo-controlled trial or a multi-arm dose-finding study. It could be an open-label design supported by external or virtual controls, The whole point is flexibility and efficiency so that we can meet the field where it is and keep pace as it evolves. In MyMatch, we have a common screening process. This is a single screening process. It is based on a common screening protocol, it's a repository protocol. In addition to matching participants to respective trials, it also serves a bigger purpose of creating a feed forward and a feedback loop between preclinical and clinical research. What I mean by that is the screening protocol creates a shared resource of clinical data, biosamples, genetic information. We generate stem cell lines from participants that researchers can then use to keep moving ALS science forward. It's one way we can connect from bedside back to bench. We always talk about bench to bedside, but this is really bringing it from bedside back to bench. We have the contractual safeguards and data sample sharing consents and data identification. All of this makes it possible for us to support ALS research and discoveries that go well beyond any one single clinical trial in MyMatch.
I see. And you've already touched on this a little bit, but the program is called MyMatch. What is meant by matching?
That's a great question. The ALS MyMatch program is built on the principles of match trials in oncology, where a central trial infrastructure and, as I mentioned earlier, a single screening process prompts timely matching of patients to appropriate trials. This really accelerates a participant's trial entry and access to experimental therapies at a site closest to them. They're early phase studies, they're not confirmatory or clinical efficacy trials. We're looking for signal pertaining to target engagement or dose finding, or even a simple question of if a drug is administered by mouth, does it really cross the blood brain barrier and enter the CSF or CNS? Does it enter the brain? We meet the ALS field where we are today. ALS is also a field that's really evolving very rapidly. If you're positioned in MyMatch to evolve nimbly with the field, whether it is identifying subgroups of ALS individuals or integrating new information that we're learning from biomarkers, genetic testing, etc. People often ask if we have as many biomarkers in ALS to do matching like oncology does. I want to take a moment to clarify that it's not about the number of biomarkers or having a wide array of ALS biomarkers, but it is about utilizing genotypes and available biomarkers, whether it's related to ALS, whether it's a neurofilament light, or whether it's related to drug mechanism itself directly, to most efficiently identify subgroups of individuals in the trial where early treatment effect signals can be detected and the drug developers and the clinical trialists can make further go, no go decisions for drug development. For example, sometimes trials may enroll based on a single genotype information. Maybe it is a trial only for C9 ALS or SOD1 ALS, or maybe only for people who have certain inflammatory biomarkers or certain fast or slow progressors based on neurofilament light. Or it could be a combination of maybe SOD1 C9 cohorts, maybe with a certain threshold of NFL or other drug mechanistic biomarkers, plus phenotypic characteristics. That's the matching process.
That makes a lot of sense. And I know that you have already been involved and completed the Acacia trial, which was an early phase clinical trial of Jacobson in people living with ALS. Can you tell us a little bit about that experience?
Certainly. Acacia trial was our first MyMatch trial that we launched in spring of last year. It's been such a fantastic experience working with a really dedicated, hardworking, and passionate team of researchers and coordination center team at Mass General. But each of our MyMatch trials are named after a tree in alphabetical order. So our first trial was Acacia, and we are very soon we are gearing up to launch our second trial, the Banyan trial, and we hope that we're able to grow a forest of trees through MyMatch program. Going back to the Acacia trial, we tested a repurposed drug in Acacia, that's digoxin, and this was an open label trial. This trial is a great example of how scientific discoveries made in the lab could have a pathway to efficiently and quickly bring to clinic through the MyMatch program. This trial also pushed boundaries on what's possible in terms of trial execution and enrollment speed in ALS. The work that was done by two Harvard researchers, Christine Marcus and Brian Wenger, really brought forth the digoxin as a repurposed drug into ALS clinical trials through MyMatch program. Christine and Brian have done a series of really elegant experiments in human-induced pluripotent stem cell-derived neuronal cell lines, or iPSC cell lines, followed by in vivo ALS mouse experiments. They tested over 2,000 unique compounds in two separate drug screens in these iPSC models. One was derived from C9 ALS individuals and another from sporadic ALS individuals. Both screens converged on the same class of drugs, cardiac glycosides, that showed neuronal cell survival and neuritic growth protection in these cell lines. Digoxin is a cardiac glycoside that's been on the market for probably half a century, and it's been used in heart failure. Leveraging this preclinical data, we designed a 6-month-long open-label trial across 3 sites in the U.S. Timeline-wise, we worked at lightning speed and launched this trial in just about 3 months. Just to provide context, a typical clinical trial may take anywhere between 6 to 9 months to launch, maybe longer in some cases. Once we launched the trial, we hit our targeting enrollment of 40 participants in just about 4 months. And again, to provide historical context, ALS clinical trials enroll at the rate of about 0.5 participants per site per month. And in comparison to that, this particular trial enrolled at the rate of 2 to 6 participants per site per month. It's almost a 4 to 12-fold higher than historical averages. The trial also showed a really impressive retention of participants for all outcomes, and especially for procedures like lumbar punctures, because it's more than minimally invasive. And we were able to achieve more than 85% retention for all lumbar punctures and CSF collections for biomarker testing in this trial. And all of this was possible because of this fantastic collaboration and partnership with each of our sites, our patient navigators, our site-facing staff, research access nurses. It's just been simply amazing. I'm very, very grateful for everyone who's been working on this trial. And now we're gearing up towards the final closeout, and we're hoping to have the results by early summer this year. Fingers crossed.
That's tremendous, and it's very encouraging to hear about how the trial went. Myself, I'm primarily a neuromuscular specialist and an ALS clinician, so I want to get your thoughts on how we bring this into the clinic. And of course, many of our patients are interested in participating in research for many different reasons. How do you think we should best talk to them specifically about these early phase 1 and phase 2 clinical trials and their importance?
Thank you for that question. At the Mass General ALS Clinic, we routinely integrate research talk into clinical care formulation discussions. Ryan, I'm pretty sure you're probably doing the same thing as well, and many ALS clinicians do this. And we all recognize that every patient is different, and it's really important as we speak about research to patients to align to their values, their knowledge, their expectations, and also balance it with their lifestyle demands and practicalities of research participation. Can they travel? Do they have childcare support? So many factors go into research participation decisions. When I'm talking about clinical trials, especially early phase clinical trials, I probably would start with walking a patient through what does clinical trial pathway look like. And I might say something like, this is a pathway through which all FDA-approved treatments are tested in a systematic manner. And every medication that's ever been approved in the US has been approved because there were clinical trials at some point in time, and there were phase 1, phase 2, phase 3 clinical trials, and some patients participated in these trials as participants that ultimately led to these medications' approval. I still remember when tofersen trial was initially launched, we had no idea when we launched the first phase 1 trial whether it would work or not and what dose would work. But we're so grateful to everyone who took the chance and participated in that first-in-human early phase trials that really contributed a very important data to the trial that formed the foundation for all later phases of testing. And then 7 years later, tofersen was approved. And tofersen is, or Qalsody, is one of the most effective therapies we have in ALS to date. But at the time that we launched the phase 1 trial, we had no idea it would work. And I mention this to patients because every trial we launch, we have no idea if it will work or not. But we launch this with the hope that it would work because we do need therapies for ALS. And we hope that every trial we're launching is that one step closer, bringing these experimental therapies towards that finish line of an FDA approval. And hopefully one day it becomes a standard of care therapy for ALS. And when I talk about the different phases of testing, I emphasize on the early phase testing as so critical to know how a drug actually works, what it does, what's the right dose, and where does it really work, in what type of subgroups of ALS population before we bring it into large 400, 500 participant phase 3 placebo-controlled clinical trials. Because the chances of those later phase trial successes are really relying on identifying and bringing forth the right therapies from early phase testing at the right doses to the right patients and at the right time window of their illness. And that's the importance of early phase testing. And we use a lot of biomarkers to get those answers.
Well, that sounds wonderful. So, how do you think we can deliver this level of care and this level of research to more people living with ALS? What are the key ways to increase access to clinical trials and the number of patients participating?
That's such an important question you ask. It was a 2008 paper that suggested that only 10% of ALS patients participate in research. And this is very different compared to some of the cancer examples where, like, for example, pediatric oncology, where over 90% patients have trial participation opportunities. And clinical trial participation is so important to accelerate therapeutic discovery for ALS. And I want to also mention that clinical research engagement shouldn't stop and doesn't stop with clinical trial enrollment alone. Other forms of clinical research, whether it's natural history studies or observational biomarker studies, digital studies, are all equally important to accelerate therapeutic discovery for ALS. There are reasons why we're not as advanced as some of the oncology fields in terms of providing access to clinical trials. On one side, it could be awareness, it could be access, it could be availability. These are important issues, but the field is actively trying to tackle these with creative solutions. For example, All ALS is a nationwide study that's funded by the NIH through Act for ALS, and this is a natural history study. This study has a fully remote cohort with home blood draws, and this is purely meant to increase access and engagement. Similarly, Clean had an expanded access program that utilized a telehealth clinic as their site and operationalized the study as a fully decentralized study across all 50 states. These were all creative solutions for improving access to clinical research. But more broadly, I do think that integrating research into clinical care systems is the direction we need to head towards, and we are heading in that direction. And this is really important for seeing those therapeutic breakthroughs. And going back to the pediatric oncology example that I mentioned before, I think really integrating research into clinical care is where pediatric oncology field saw huge successes. And in pediatric oncology, there are federally funded centers of excellence. And so more than 60 to 70% of pediatric cancer patients enroll in trial, more than 90% enroll in some form of clinical research. That very high level of research participation and collaboration that was established through creating these cooperative groups in oncology really led to major survival improvements in several cancers. That's the hope we have for ALS when we hope for integrating research into clinical care. And part of the reason is not just about the trial. Part of the reason is that these clinics routinely collect biosamples, genomic data. Everybody that's working in the clinical realm is also trained to collect research data and samples to allow for workflows that brings in precision medicine approaches. And that's the direction we should be taking in the ALS field as well.
That makes a lot of sense to me as a clinician as well. And I think that investigators and doctors in the clinic will agree that ALS remains daunting. It remains challenging. There's so much work to be done. For you right now, researching and practicing in the field of ALS, what are your greatest sources of hope looking forward to the next decade?
I'm very encouraged by how the field is evolving all around, from preclinical to clinical trial design, and also the community, how collaborative and interconnected and interdisciplinary ALS research is becoming, and how we're incorporating AI and technology into drug development. I'm super excited by all these developments in ALS. And it's also really wonderful to see all the traction in ALS advocacy, all the funding support from philanthropy, foundation, federal sources for promoting ALS research. There's still a lot of work that needs to be done, but we're heading in the right direction. I think this collective momentum is so powerful, And I'm hopeful that we will see some real breakthroughs. And this is really coming from a place of grounded hope after seeing successes with tofersen, that we're not too far from changing ALS from a universally fatal to a potentially livable disease.
I share that hope with you 100%. And I am so excited to see the direction of your research and to continue to learn from you and to really join with you in this endeavor. Thank you so much for joining us on the podcast today.
Ryan, thank you so much for having me. This was really fun chatting with you today.
That was Dr. Suma Babu of Harvard Medical School and Mass General Brigham, interviewed by Dr. Ryan Jacobson of Rush University. ANA Investigates is the official podcast of the American Neurological Association. The show's producers are member of the ANA Education Innovation Committee, chaired by Sarah Nelson and Sharon Ellen Govan. I'm Ifra Zawar, the podcast host and executive producer. Our production team this month included Ryan Jacobson and Jen Hurley. You can listen to the past episodes of the show, subscribe to us so you never miss an episode, and access all of the ANA's educational program on the Education tab at myana.org. That is it for ANA Investigates. See you next month.